In this portion of our discussion of muscle tissue, we'll discuss smooth muscle tissue. This is a unique tissue that has some very interesting features and is often overlooked. Smooth muscle cells are shaped like hot dogs with pointy tips at either end. We call this shape fusiform, so pointed on the end and then wider in the middle. Smooth muscle cells contain only one nucleus, and they're very, very small. If you remember when we looked under the microscope, you could see many smooth muscle cells on high power. But when you looked at skeletal muscle or cardiac muscle on high power, you could generally only see part of a few muscle cells. So smooth muscle cells are very small. Smooth muscle cells have no striations, so they do not have the orderly arrangement of myofilaments that skeletal muscle and cardiac muscle has. There are no sarcomeres or Z-discs. Those Z-discs are part of that orderly arrangement of fibers. Instead, we have thin filaments that are attached to dense bodies. So dense bodies are basically just like attachment points for those thin filaments, so those filaments have something to pull on. And those dense bodies are scattered throughout the sarcoplasm, that's the cytoplasm of the muscle cell, and those dense bodies are attached to the sarcolemma, the plasma membrane of the muscle cell. The sarcoplasmic reticulum is scanty. That means there's very little of it in smooth muscle cells. And smooth muscle cells have no T-tubules. All of the calcium for smooth muscle contraction comes from the extracellular fluid. If present, nerve supply is going to be autonomic, which means that the nerve supply is from our involuntary nervous system. We're not going to consciously control smooth muscle. It is going to be controlled unconsciously. This nerve supply releases either acetylcholine or a neurotransmitter called norepinephrine. We'll talk more about these two neurotransmitters as we move into the nervous system and how our autonomic nervous system works. There are two types of smooth muscle. We have single unit or visceral smooth muscle, where all of the smooth muscle cells are connected together. So we have more than one muscle cell, but they function as a single unit. They are electrically coupled by gap junctions, just like the cardiac muscle cells are. This allows the electrical signal to move from one muscle cell to another muscle cell, so that all of them contract together as a unit. We find this type of smooth muscle in most blood vessels and in the viscera, our digestive tract, as circular and longitudinal muscle layers. Remember, when we saw smooth muscle in lab, it surrounded the intestines of a little critter. So we were actually looking at the circular and longitudinal layers of the visceral single unit smooth muscle. In the viscera, the smooth muscle all contracts in a coordinated fashion in order to move food through the digestive tract and to churn food within the digestive tract. We also have a second type of smooth muscle called multi-unit smooth muscle. In multi-unit smooth muscle, the nerve fibers that come and branch and synapse or join with the smooth muscle, those nerve fibers terminate on individual myocytes. So you can see that up here on the single unit, we have a nerve fiber on just some of those myocytes. And then we're going to transfer the signal through the different muscle cells in order to communicate and have all of those cells contract as a unit. Down here on the bottom, we have a nerve fiber that connects to every single myocyte. So that's multi-unit smooth muscle. This type of smooth muscle exhibits independent contraction. I can make these muscle cells contract by sending a signal down this neuron. I can make these muscle cells contract by sending a signal down this neuron. I don't have to contract all of them at the same time. We find multi-unit smooth muscle in the largest arteries, in the iris or colored part of your eye, in our pulmonary air passages, and in the erector pili muscles. So if you remember those little muscles that make our hair stand on end, those are multi-unit smooth muscles. So they don't all contract together. You may get goosebumps on one part of your body, but not on your whole body at the same time. Smooth muscle also works a little bit differently than skeletal and cardiac muscle. Smooth muscle is involuntary and can contract with or without nerve stimulation. So we just looked at two types of smooth muscle that contract with nerve stimulation. However, without nerve stimulation, we can have other things that trigger smooth muscle to contract. Some hormones will trigger smooth muscle to contract. Carbon dioxide can trigger smooth muscle to contract. A low pH or acidity, stretch of the smooth muscle cells, or oxygen deficiency can all trigger contraction of smooth muscle. We also have specific cells within the GI tract that are autorhythmic. They just spontaneously depolarize every so often. We call these pacemaker cells, just like the cells in the heart. With nerve stimulation, we can have autonomic nerve fibers that go to the different types of smooth muscle. And in these autonomic fibers, we can have bead-like swellings or varicosities. So varicose veins are actually just a swelling in a vein. So a varicosity is just a swelling, kind of a round bead-like swelling. These bead-like swellings in nerve fibers that go to smooth muscle contain synaptic vesicles with neurotransmitters. Synaptic vesicles are a certain type of vesicle that are found at synapses that get exocytosed so that we have the neurotransmitter release onto the muscle. And these varicosities can stimulate multiple myocytes, like we saw in the last slide. So here's another picture of smooth muscle with an autonomic nerve fiber, and you can see the varicosities, these bulges, and in these varicosities we have synaptic vesicles that will be exocytosed so that the neurotransmitters can be released onto all of these smooth muscle cells. So this is what we have when we're looking at single unit smooth muscle, especially smooth muscle in the viscera or digestive tract. Features of contraction and relaxation in smooth muscle are unique to smooth muscle. This is one of the things that makes smooth muscle really interesting. As I said before, the calcium triggering the contraction is from the extracellular fluid. These channels that let in calcium are triggered to open by voltage, hormones, neurotransmitters, or cell stretching. So these are the same things that cause the cell to contract, because calcium coming in is the first step in cell contraction. These calcium ions bind to a regulatory protein called calmodulin. When calcium is not present, calmodulin prevents contraction. When calcium activates calmodulin, calmodulin in turn activates an enzyme called light chain myokinase. This light chain myokinase activates the motor on myosin. We call that myosin ATPase. So it basically starts up that motor. When that motor is started, we have a power stroke. So the myosin ATPase pulls on actin and makes the cell smaller. It pulls the two cell membranes closer together. This power stroke occurs when myosin hydrolyzes or breaks apart the ATP. These thin filaments pull on intermediate filaments attached to dense bodies. We talked about those dense bodies as just being attachment points on the plasma membrane, and this causes the entire smooth muscle cell to shorten in a twisting fashion. So it kind of squishes up, so it shortens in length and in width as it twists and squishes together. So here's the process of smooth muscle contraction. Calcium is let in from the extracellular fluid. As it comes in, it activates calmodulin. So we have inactive calmodulin. When the calcium binds to it, so calcium is bound right here, it activates calmodulin, activated. And notice that calmodulin changes shape when it's activated. When that calmodulin is activated, then it can then turn an inactive kinase into an active kinase. This active kinase can then turn on the myosin ATPase. That's our motor that pulls. Once the motor of myosin has been turned on, the myosin will pull on the actin, the thin filament, and shorten the muscle cell. Notice that in smooth muscle, these myosin heads are pulling this direction, whereas the myosin heads on the other side of the myosin molecule are pulling in the opposite direction. What this does is it shortens the cell in both directions. So this is a little bit different than how skeletal muscle works. And when we look at skeletal muscle, you'll see that all of the myosin heads pull in the same direction. So in smooth muscle, the myosin heads pull in opposite directions. So that's an important difference between smooth muscle contraction and skeletal muscle contraction. Smooth muscle is very unique in that contraction and relaxation are very slow in comparison to both skeletal and cardiac muscle. In general, it is about 30 times slower to contract, so it takes a long time to contract. It's not very quick. This is because that engine, the myosin ATPase, is a very slow engine. It doesn't work very quickly. So it's a different variety than what we see in skeletal muscle. So we have a slow ATPase and slow pumps that remove calcium. So not only is contraction slow, but relaxation is slow. In order to make a smooth muscle relax, one of the things that has to happen is we have to remove calcium, because calcium is the trigger for contraction. So the pumps that move calcium out of the smooth muscle cell back into the extracellular fluid are very slow. Our pumps in skeletal muscle and cardiac muscle move much faster than the pumps in smooth muscle. One of the other really amazing things about smooth muscle is that it uses 10 to 300 times less ATP to maintain the same amount of tension or force. So that's pretty incredible that we can maintain the same amount of force with less energy. So smooth muscle is pretty efficient. This tension is important because it keeps our arteries in a state of partial contraction. We call this vasomotor tone. If our arteries just completely relaxed, all of our blood would fall to our feet because of gravity, and we would pass out. It's not usually a good thing. So we want to maintain enough vasomotor tone that we force blood up to our head. So this is an important feature of smooth muscle within our arteries. The current theory is that there's a latch bridge mechanism that maintains that contraction. So once the myosin head attaches to actin, it holds on and forms a latch bridge. scientists are still gathering evidence to determine whether the latch-bridge mechanism theory is truly how smooth muscle can maintain the same force with less ATP use. This is an area of research. It's very difficult to research, and it's an interesting area, but they have not fully figured out why smooth muscle is able to do this at this point. So when smooth muscle contracts, we've got some important features here. We have the intermediate filaments. These intermediate filaments, these are the actin filaments. We have dense bodies. You'll notice these both at junctions between the intermediate filaments and near the cell membrane. We have gap junctions, so this would allow this cell to stimulate its neighboring cell. And we have calveoli, which are like just small indentations in the cell. They're like, yeah, I thought about putting a T-tubule in, but all I got was this indent. So those are areas where the smooth muscle cell has a little bit of increased surface area, so we can let more calcium in. So the gap junctions connect to another smooth muscle cell, whereas the calveoli allow calcium to come in from the extracellular fluid. When a smooth muscle contracts, it contracts in length, and it's a little hard to see from this, it will contract in width as well. So this smooth muscle will contract both ways, and it contracts in a twisting fashion, so you kind of get this little S-shaped, squished cell. And you can see how the nucleus gets all squished up inside the cell instead of being long and skinny like it is in a relaxed smooth muscle cell. So this is a little deceptive because this cell up here not only shortens in length, but it also gets skinnier as it shortens. So pretty much the whole thing squishes like a little slug. One of the characteristic responses to stretch by smooth muscle is that we can use stretch to cause contraction. In smooth muscle, stretch can open mechanically gated calcium channels. So remember, we have those channels. They can be opened by chemicals. They can be mechanically gated, respond to stretch, or they can open in response to voltage changes. So these specific channels open in response to stretch, and they let calcium in, which causes a muscular response or contraction. One of the areas we see this is in the esophagus. That's the tube that leads from your mouth to your stomach. When you begin to swallow, and that clump of food enters the esophagus, it stretches out the smooth muscle within the esophagus. So the walls of the esophagus have smooth muscle. When those smooth muscle cells are stretched by the food, they contract, and that pushes the food farther down your esophagus. Then the next group of cells are stretched, contract, and continue to push that food down into your stomach. So this allows the process of swallowing. Once you start swallowing, your body, your involuntary nervous system takes over, and those smooth muscle cells will finish that swallowing response because of that stretch response. There's an important response that's required for filling of hollow organs like your urinary bladder. This is called the stress relaxation response. In contrast to your esophagus, the smooth muscle cells in your esophagus will contract when stretched. In your bladder, when those smooth muscle cells are stretched, they briefly contract and then relax. And this allows your bladder to stretch and fill. If we had the same response in our bladder that we did in our esophagus, every time we got urine into our bladder, those smooth muscle cells would contract and we'd pee just a little bit. That would make things very messy. So it's really handy that our bladder is able to contract slightly and then relax in order to fill. However, when our bladder is completely full, we would like to be able to empty it. So these same cells must contract forcefully when they're greatly stretched. And the way that these smooth muscle cells can do that is because the thick filaments have heads along their entire length. There's no orderly filament arrangement, so there's no sarcomeres, no Z-discs, no striations. They're able to contract in all directions at once when stimulated. So there's an important term that goes with this process. It's called plasticity. Plasticity is the ability to adjust tension to the degree of stretch so that the empty bladder is not flabby and the full bladder is not contracting. So plasticity is the ability to adjust the amount of stretch and contraction coming from those smooth muscle cells that surround the bladder. So plasticity is an important term in the urinary bladder filling and emptying. And that finishes up our discussion on smooth muscle cells. Next, we're going to move on and talk about our voluntary muscle. That's going to be our skeletal muscle.